Hello everyone, welcome back. In this video, let us discuss about Genomic Library. We can also call it as Gene Library. Generally, in our college, we used to go to library to refer a book, right? Because library is a place we can find huge collection of books, right? When it comes to Genomic Library, here we can find a set of genes that constitute the entire genome of an organism. So Genomic Library is a collection of plasmid clones or phage lysate which contain recombinant DNA molecules. Together, the individual DNA inserts in the collection of molecules represents the entire genetic information of an organism. Now let us discuss about the construction of Genomic Library. Look at the picture. First, we need to isolate the chromosomal DNA of an organism of interest. Whether we go with the microorganisms or human beings, whatever the organism you choose, first step is to isolate the chromosomal DNA of an organism. Then this chromosomal DNA is partially digested with the help of restriction enzymes, which results in the larger DNA fragments. So look at the picture. This is the gene A, B, C and D. After partial digestion, we got the DNA fragments carrying each gene A, B, C and D, right? Don't go with the complete digestion. If we go with the complete digestion, that results in the DNA fragments which are too heterogeneous in size. To avoid that problem, we generally go with the partial digestion. For partial digestion, we prefer the use of restriction enzymes that can recognize either 4 bases or 6 bases. Generally, 4 bases occur for every 256 base pairs and 6 bases occur for every 4096 base pairs. Now, after enzymatic digestion, what we get? We get different DNA fragments, right? Those DNA fragments are separated with the help of a technique called electrophoresis. I already made a video on electrophoresis. I'll provide you iCard. Please go through that video. After this step of electrophoresis, we add synthetic linkers to the fragments. Okay, so these linkers are attached at the ends of the fragments, which results in the cohesive ends. With the presence of these cohesive ends or sticky ends, it is possible to pack them into phage heads, which is nothing but in vitro packaging. So here these phages are carrying recombinant DNA molecule, right? See, after the isolation of this DNA fragments, they are now joined with the lambda DNA pieces with the help of an enzyme called DNA ligase. After the formation of the recombinant DNA molecule, as they carry cohesive ends at their ends with the addition of synthetic linkers, it is very easy to pack them into a phage head. These phases constitute the genomic library. See here, each phase is carrying a respective gene. This phage is carrying A gene, B gene, C gene and D gene. So they together constitute the entire genome of an organism. And now these phages are ready to infect the host organism. All these phases are carrying the recombinant DNA molecule, right? How we made a recombinant DNA molecule? The foreign gene is inserted into a vector, right? With the help of the synthetic linkers, we made a cohesive end. So with the help of those cohesive ends, we have packaged them into phage heads by a process called in vitro packaging. See the step here. Joining to lambda DNA pieces followed by in vitro packaging. So this foreign DNA is inserted into a vector and then they are packaged into a phage head by in vitro packaging, and now these phases are ready to infect the bacteria, that is E. coli. For suppose if I want to identify a particular gene and I don't know which phase is carrying that particular gene. For that reason, I go with the screening methods and I use certain hybridization methods in order to detect the gene of interest. Now let us see how to screen gene of interest. Look at the picture carefully. See, these are the lambda virions harboring foreign DNA. Now they are ready to infect the host organism. As I told you just now, if we want to identify a particular gene of interest, we go with the screening methods, right? So here the amplification of the gene will take place by infection with E. coli. Genomic library in lambda phase. and this is the culture plate containing the colonies of clone of organisms. Now what we do? We are going to add a dilute suspension of these recombinant phages to a culture of bacteria, and these phages are now going to infect the bacteria, forming a plaque. See, this is the surface view of the culture plate showing the bacterial colonies. Now we take a nitrocellulose paper and we gently press on these colonies. So we are going to make a replica of this master plate. Whatever the colonies which are present over here will be sticked onto the nitrocellulose paper exactly at the same location. So it will be now used as a replica of master plate. Whenever these bacteriophages infect the bacteria, DNA will be released out, and that DNA will stick on to this nitrocellulose paper. Once we go with the alkali treatment, the DNA which is present over this nitrocellulose paper will get denatured. See here, the DNA got denatured, making it accessible for hybridization with radio-labeled probes. We use P32 radio-labeled probes. So look at the picture carefully. This nitrocellulose membrane with the denatured DNA is treated with radio-labeled probes. These probes will go and bind with the DNA sequence if they both are complementary to each other, right? When they are subjected to autoradiography, we can identify the spots. This spots indicates that the hybridization has taken place between the probe and the DNA sequence. So this autoradiography reveals the position of spots carrying the recombinant DNA molecule. Now these plaques are picked up from the master plate which are grown separately. This is all about the genomic library. In my next video, we will discuss about the cDNA library. Thank you.